Hello, my name is David Girata, and I am an undergraduate researcher in Dr. Levine's Physical Genomics Laboratory. My project is titled Unbiased Characterization of eccDNA and Clinical Applications. So eccDNA, or extrachromosomal circular DNA, is a type of DNA molecule that exists outside of what we normally conceive of as the linear genome. A picture in the bottom left here has a representation of what this looks like in the cell. So we have chromosomes composed of what we call linear DNA, and then we have the circular DNA molecules maintaining some topological characteristics, such as being wrapped around histones that also exist within the nucleus, but they vary both in size and sequence depending on the type of cell. So they can go from very small, such as hundreds of base pairs, to very large, such as millions of base pairs. It also means that not all of the eccDNAs in each cell are going to be the same. That is, the eccDNA repertoire in a skin cell is not going to be the same as the repertoire in a liver cell. However, a special subset of these eccDNAs has been observed to give cancer cells, some cancer cells, chemotherapeutic resistance. How this happens is by a process called chromothripsis. So chromothripsis is a genome shattering event in a cell that makes it cancerogenous. So the linear DNA, the linear chromosome in the cell, is turned into linear fragments, into smaller fragments. And as the cell is trying to cope with this genome shattering event, instead of incorporating a linear fragment back into the genome, it will stitch it end to end as an error, and it will create a circular molecule. This circular molecule will have a new novel DNA sequence that will then give rise to a novel protein that may give this new cancer cell chemotherapeutic resistance. These circles that have these proteins that give the cell chemotherapeutic resistance tend to be in the larger size range, so about hundreds of thousands of base pairs. And current biophysical techniques are unable to efficiently purify these very large circles. So as a result, we have developed a new technique that is able to purify these really large eccDNA circles while keeping them topologically intact. This technique basically relies on embedding the total DNA from a cell nucleus in a hydrogel matrix, processing it through a patented centrifugation process. This will yield a solution of topologically intact eccDNA molecules. And the topology of these eccDNA molecules is important because it gives an indication of whether they were part of the genome at one point or not. These circles can be then sequenced, and we can observe these novel DNA sequences that give cancer chemotherapeutic resistance, as well as see, for example, some oncogenic sequences such as MYC being in these circles and being amplified. So through this process, then, we are able to appropriately characterize, and as we continue to process more DNA samples from different types of disease models, we are better able to understand the roles of these circles in the stability of the genome. So the genome will not stay the same throughout the lifetime of the cell. It will change because of the existence of these circles. So some of these eccDNAs will be excised from the linear genome. They will be reincorporated. The genome is not static, basically. And having a technique that is able to purify all types of circles in different sizes and sequences will allow us to properly understand the mechanisms through which these eccDNAs are formed in all types of disease models, as well as in normal cells. Thank you.